Good evening, everybody, and welcome to our webinar titled Research 101, Driving Progress Through Clinical Trials and the ADPKD Registry. My name is Paul Scribner. I'm VP of Community Programs and Education at the PKD Foundation, and I'll be moderating today's presentation. So we are excited to be joined today by leading experts in PKD research and a patient advocate who will talk us through research basics, the role of the ADPKD Registry, and how clinical trials advance new treatments. Their insights show how research improves care, supports informed decisions about participation, and underscores the essential role of patients and families in driving progress toward a cure. Now I'd like to introduce today's speakers. Chris Chen has studied PKD for more than 20 years, inspired by his family's experience with the disease. He holds a degree in genetic biology from Purdue and a PhD in molecular and human genetics from Baylor, and completed a postdoctoral training at Yale and the Mayo Clinic, which was focused on RNA biomarkers in PKD. At the PKD Foundation, he guides research strategy and advances programs aimed at accelerating new treatments for PKD. And he's PKD's Vice President of Research Programs, as it says there. So Dr. Seliger is an Associate Professor of Nephrology at the University of Maryland and has been leading PKD clinical PKD clinical investigator for 15 years. He has directed key PKD research cores, chairs the clinical subcommittee of the NIDDK funded, that's a federal agency funded PKD research resource consortium, and plays major roles in multiple ADPKD clinical trials. He also serves as the PKD Foundation's ADPKD Registry advisor on the advisory committee and scientific advisory committee. Tasha Rasper was diagnosed with ADPKD at age 4. Inspired by her dad's journey, has been actively involved in PKD research since 2008. A dedicated PKD volunteer, she helped establish a PKD Center of Excellence and Idaho's first PKD Walk Your Way event. After a double nephrectomy and a preemptive kidney transplant from her husband last year, she remains committed to advocating for PKD research. So at this time, I would like to turn things over to Chris Chen to get us started.
Great, thanks, Paul. I've been tasked with trying to describe what is PKD research and help understand the basics and why research matters. Slightly counterintuitive. I'm not sure if I'm actually the best person to ask what is research. I think when I asked myself this question, the first answer that I had was that for me, thinking about research every day, and research is really life. And I understand that this may not be the perspective for everybody on the call, but also recognize that I think intuitively, everybody has a pretty good understanding of what research really is. So I'm going to take a minute and really maybe provide a little bit of framework for how I think about biomedical research in particular. And for me, it's really just understanding the body in its healthy state. How does the body function in a healthy state? How do the organs operate? How do cells interact with each other? How does a cell operate within the cell? And then comparing that to the disease state. So what is happening, again, whole body, and then whole organ, between tissues and within the cells. Using that information to really advance different types of prevention, diagnostics, and treatment. Specifically within PKD, we're looking at what is happening within a healthy kidney and comparing that to what's happening in a cystic kidney. How are cysts developing and initiating? What is causing them to grow? And what is the impact on those growing cysts on the remaining tissue within the kidney? And then we can take that information, again, and apply it specifically to preventative or diagnostic or new treatments for PKD. So and try to better understand the full spectrum of research. Research is kind of bucketed into three groups. There's basic research, applied research, and clinical research. Basic research is kind of the first step that I mentioned before, trying to understand how healthy kidneys operate, and then going from there to understanding what's happening in the disease state. Applied research is a little bit of the opposite. It's really going from trying to understand what's happening in the disease state and trying to see if we can use that information to either prevent the disease from progressing or for developing treatments that might revert the kidney back to a healthier state. So in the case of therapeutics, these are tested in cell culture and petri dishes. We identify things that are working in cell culture and then move them on to preclinical models, most commonly the mouse model. We have very good mouse models for PKD that mimic the exact mutations that occur in some patients and develops cystic phenotypes within, or cystic characteristics within those animals that we can test new compounds in to see if they're working. If they're shown to be effective in our preclinical models, then we move them into clinical testing, where we move them into patients to see if we can get the evidence to show that they work and should be clinically approved for the entire patient population. So I want to spend some time on clinical trials specifically. I think many are aware that clinical trials are segmented into different phases, although I think trials can get very complicated. I want to give kind of the basic framework for that most clinical trials are designed based off of. So the first phase of clinical trials are safety trials. They seek to answer the question, is an investigational medication or treatment safe? Some of the other things that they test are really the kind of the metabolism through the body. Where do these drugs go in the body? How long do they last there? How long and how are they degraded from the body? And then also looking at different doses of the drugs, trying to identify what is the range at which the drug use can be safe. These trials are pretty small, usually less than 100 people. They last in the range of a couple of months, and about 70% of trials that enter phase 1 are shown to be safe and move into phase 2. So phase 2 studies are efficacy studies with the intent of answering the question, is the investigational medication or treatment effective in treating the targeted condition? Just like phase 1 and actually all phases of clinical trials, they're going to monitor for safety. With regard to dosage, we're looking at identifying the most effective safe dose that can be used in patients. And these studies are a little bit larger, so generally between 100 and 300 patients. The duration of the study is significantly longer, usually 1 to 2 years. And fewer studies actually progress and show efficacy. So about 30% of studies that enter phase 2 move on to phase 3. Phase 3 studies are confirmatory studies. You can almost think of them as larger scale phase 2 studies. Again, they're going to be looking at efficacy and they're going to be looking at safety. But more specifically, they're going to compare treatment to the standard of care. So what are the best care for patients currently, and is this potential new therapeutic an improvement upon that? These studies are much larger. So you're looking at between 300 and 3,000 people. The duration is typically between 2 and 3 years. And the kind of the success rate for phase 3 studies is in the range of 50%. So the data for phase 1 through phase 3 studies is generally put together if it's shown to be confirmed to be efficacious and put together in a data package that's presented to the Food and Drug Administration, the FDA, who reviews the data package and determines whether or not to approve it for a given condition. There's actually a phase 4 clinical trial. This is actually after the approval of a drug. And it monitors both the safety and benefit of the drug in the long-term timescale. So this is actually one of the ways that the registry, the ADPKD Registry, can be quite beneficial for research understanding of how well the drug is working long-term and how safe it is. By participating in the registry, for example, those who have been on tolvaptan were able to track the safety and efficacy of tolvaptan users long after it's been approved. So part of what I want to do today is kind of tee things up so that for Dr. Seliger's presentation, so I want to cover some basic concepts within clinical trials. The first is institutional review boards, or IRBs. These are independent committees comprising of physicians, scientists, and lay members of the community who evaluate trial protocols, which is really the study designs of the clinical trials, to ensure that the benefits of those trial designs and the protocol outweigh the risks, and also to ensure that the studies are designed in an ethical manner. So just two things to highlight here. First is that this is an independent committee. So you can imagine many of these clinical trials are designed by industry. The committee that's reviewing these clinical trial protocols is independent and not influenced by industry. And secondly, that it's not just experts, not just the clinicians and the scientists experts, but also lay members of the community that are doing the evaluation. The second concept I want to introduce is informed consent. This is the process by which participants are made aware of key facts about the protocol before deciding whether or not to participate. So these include the purpose of the study, the duration and the procedures, and really just to make sure that there's nothing being hidden from patients in terms of what's understood about the study and how it's going to be executed. So they can really have a full understanding before deciding whether or not to participate in the study. In terms of the design of the study, there are a couple design elements that are kind of gold standards for ensuring the greatest power of the data. The first of those is randomization. So the process of randomization is to generate unbiased assignments of participants in a study to either the treatment or the placebo groups. So the placebo group is another word for like a control group. These are patients that are going to undergo the same procedures as those who are getting the treatment, but there is no actual test article that's being provided to the people in the placebo group. So they're really the comparative group to the treatment group that is going to be used to show that the treatment group, whether or not it has efficacy or not. The second element of the study design to really highlight is blinding. So this is the process by which either participants in the study or physicians do not know whether or not the patient is receiving the treatment or assigned to the treatment group or the placebo group. And again, this is preventing bias of either the participants from the kind of data that they're reporting, or for physicians who are collecting the data or reporting the data from having any preconceived ideas of how the particular response to the treatment is supposed to be. And I want to focus a bit more on ways to get involved in different steps and parts of the research pathway. The first is how to get involved in basic research. One way is through tissue donation. For those patients who have had nephrectomies, the PKD Foundation does have a mechanism by which you can donate your kidney to research. In addition to just the kidneys, it's also possible to donate blood and urine samples for research. The foundation doesn't have its own pathway for this, but many of the centers of excellence have their own biobanks for this purpose. So I would encourage you to talk to your nephrologist about whether or not there is an opportunity to donate tissues and biospecimens like blood and urine. It's actually going to be very important for future research to use those samples in combination with data that exists within the registry to identify what we call biomarkers, or things that you can measure from those samples that might indicate whether or not a treatment is working or how a disease is progressing. So a lot of these biomarkers that are kind of developed through these donated tissues will be used for future clinical trials. Coming back to sharing your data on the registry, another important aspect and outcome from sharing your data on the registry, in particular sharing electronic health records, is helping to establish a natural history or a normal progression for disease. Many of you may be aware that tolvaptan is not recommended for patients that are slow progressors of PKD or for pediatric patients. And that's not necessarily because tolvaptan wasn't shown to be efficacious for those patients, but it actually just wasn't tested in those initial clinical trials in those populations. It's quite a bit harder for slower progressors or pediatric patients to show efficacy. But in the future, if we have enough of this natural history data existing within registries, we may be able to, for future clinical trials, to include slower progressors of disease or pediatric patients, and then including them into those clinical trials may enable them to be recommended for potential drugs that are clinically approved. Moving on to getting involved in applied research. So the PKD Foundation, the research program, funds $2 million of research grants every year. Many of those grantees are studying new potential therapeutics. So anybody who's participating in the WALK program or just donating to the foundation is really helping to support a lot of these very early stage research projects that are looking at novel therapeutics. I also want to bring up make you guys aware of participating in advocacy efforts for research funding. So the foundation supports a group called the Advocacy Champions Network, the ACN. The ACN is a group that we use to meet with legislators and advocate for federal funding for research specific to ADPKD. For those who have participated in that process before, really the opportunity to tell your story to legislators has a significant impact on research funding. And right now, for those who aren't aware, the research funding environment is in a bit of flux, so it's more important than ever to really be advocating for federal funding for PKD research. For clinical research, I know Dr. Selger and Tasha are both going to talk at length about participating in clinical trials. I do also want to introduce the Community Reaction Panel. This is another group within the PKD Foundation that when industry partners come to us in their clinical trial design or in their drug development process, wanting to get the patient perspective, we pull patients from the Community Reaction Panel or caregivers from the Community Reaction Panel to help serve on advisory panels, advisory boards, or to take various surveys to help inform that process of drug design or clinical trial design. So I've mentioned the ADPKD Registry multiple times, and I just want to kind of summarize the different ways that participating in the ADPKD Registry can advance research. Really, one of the most important is accelerating enrollment into clinical trials. I mentioned the length of time that the phase 1, phase 2, and phase 3 trials take, and also the number of patients involved in those trials. It can be very difficult to fully enroll those studies in a time-efficient manner. And so the faster that we can get those studies enrolled, the faster that we can get these studies completed and hopefully new drugs approved for PKD. There's also, I mentioned the value of establishing natural history patterns in ADPKD so that we can enable younger or slower PKD progressors to get access to treatment. The biomarkers that I mentioned are essentially new readouts that, when paired with the registry data, help determine whether novel drugs are working. And as I mentioned, the phase 4 clinical trials that are looking at the long-term effects of drugs, that data, a lot of that data can be supplied from the registry. So I want to take a second here and maybe summarize in terms of and give a better overview of the length of time that this whole process takes. So looking at the column on the left in green, drug discovery is really reflective of the basic research phase, really target identification, looking at potential candidate compounds for small molecule designs at least. This is a process that can take 3 to 5 years. Then moving on to preclinical studies. This is another name for applied research or translational research. Again, testing in cell culture or in animal models. This is a process that can take 1 to 2 years or really quite a bit longer. Clinical trials themselves, the phase 1, phase 2, and phase 3, when you add it together, typically takes 6 to 7 years. And then the regulatory approval by the FDA, 1 to 2 years. So All in all, you're looking at 11 to 16-year process to get a drug to market from starting from really the early initial discovery phases. I just want to have one more slide to show you kind of what that timeline looks like in the perspective of PKD. So for those who are interested in the history of PKD, is really first described in 1586 in a Polish king. And it wasn't really until the 1970s that we started to see some observations of patients with PKD that helped us move closer to developing treatment. So in 1972, the observation that PKD patients have urine concentrating defects. And then about 30 years of research, basic research, really understanding the mechanism by which that urine concentrating defect occurs, and then identifying potential small molecule therapeutics that might be able to block that defective pathway. Those initial early preclinical studies of what would later become tolvaptan reached phase 3 and completed phase 3 in 2012. And then many are probably aware of the treatment, the FDA approval of our only treatment for PKD to date, tolvaptan, in 2018. So you can see that the 2004 to 2018, that 14-year span, going from initial preclinical studies to actual approval of the drug. And that's really actually accelerated by the fact that tolvaptan is a repurposed drug. It was initially approved for hyponatremia in 2008. So it didn't, for PKD, didn't have to go through that phase 1 process. So this was even kind of accelerated in the fact that it was a repurposed drug. Now, the good news is now, as we're on the precipice of going from 2025 to 2026, there are a number of large clinical trials that are in the phase 2 to phase 3 phase. And really kind of showing you where you can kind of extrapolate this back to kind of we're in the kind of where we were in 2010, but for a lot of different new and exciting drugs. Again, this is just a sampling of some here, some of which Dr. Seliger is going to present in just a minute. I know he's an expert in some of these trials themselves, and that he'll be talking about them, as well as a number of the repurposed drug trials. So with that, I am very excited to turn the floor over to Dr. Seliger.
Wonderful. Thank you so much, Chris. And it's a great pleasure to speak with everyone today. And I'm at the University of Maryland School of Medicine. This, as Chris said, this is a very, very exciting time in my 15 years of research with my colleagues at the University of Maryland. We've seen really an explosion of interest in novel treatments being tested. And so I think there is a sense of real excitement and optimism about what's coming down the pike. I just want to acknowledge that I have received research funding from a number of industry sponsors. Of course, these are the ones who are running the clinical trials that we'll be discussing, but in no way am I here to promote any one particular study over another, but I'm going through quite a few of them. So before I get to the individual studies, I do want to just address a question which many people ask me, which is, what is the point? What do I get out of taking part in a study as a participant, as a research participant, as a volunteer? And so there's sort of three ways I kind of conceptualize this. One is the potential for direct benefit. So if someone's in a study, if they are assigned, often at random, as Chris described, and then get the active treatment, and the treatment works, well, then, of course, one can benefit from their use of that medication. And sometimes there are opportunities after the study is over to continue with that, even before the drug is approved. But, of course, not all drugs are shown to work. And then even in most studies, about half of the participants are assigned to placebo. So what's their potential benefit? And that can be from more careful monitoring and measurement of their disease. And one common example is this measure of this standard biomarker of disease risk of the total kidney volume, which is often challenging to obtain from private medicine with MRIs, but is almost always measured in these studies as part of research. And so this can better inform patients and their treating physicians about the progression of their disease. And then, in my experience in being involved with many clinical trials over the last 15 years, often the greatest benefit or motivation of is really to develop and create knowledge that will benefit all patients with PKD around the world from time to come. And this is acknowledging that the development of new medications and treatments is entirely dependent on patients volunteering to participate in clinical trials. And a good example of that, as Chris said, is the FDA approval of tolvaptan, the first drug, and so far the only one approved to slow progression of PKD, which involved more than 1,000 individuals around the world volunteering to participate. So since we're talking about volunteering, I want to just also spend a brief period of here going through sort of what that means. And as Chris described, there's a process of informed consent. And I wanted to emphasize that this is a process. Often in clinical trials, we talk about the informed consent form or document, and that is extraordinarily important. But really, the document itself is part of a process, a series of communications between the research team and a person who is interested in participating. So it was shown it provides key information about the research, and then it confirms that the participant really understands the information that's being provided, and that any decision to participate is completely voluntary. And that's shown in this graph here, the sort of key components of competence and comprehension and information disclosure. So for those who choose to participate or even to begin the process, am I thinking about participating, often this is where it leads to, or this is sort of a key junction point. Okay, so then we want to sort of think about, as Chris said, alluded to, who should or does or can participate in clinical trials. And he brought up the example of tolvaptan, which was only studied in people who were at higher risk for progression. And so this gets to a general principle in clinical trials, which is particularly with new molecules or new drugs, we want to identify those who have the greatest risk of progression if they don't get a treatment, and also are at the lowest risk of side effects or adverse effects of a given medication. So that the latter part of that is based on, again, what we know about the side effects of the drug, but who's at risk can be assessed clinically and in research by a number of different processes. And some of you may have heard about these. One is the Mayo imaging class, which uses the total kidney volume based on someone's age. And this is just an example of what that looks like. So the green line is people who have the smallest kidneys for their age. And you can see over time in the x-axis, and the y-axis is the estimated glomerular filtration rate or kidney function, that the decline of their kidney function actually is not specifically different from the black line, which is people who don't have PKD. In contrast, that dark orange line on the bottom, that is people who have the largest kidneys for their age, and you can see that they can progress to kidney failure even in as little as 10 to 11 years. So this is often used to identify people who might be eligible to participate and have derived the greatest benefit. But there are other systems as well, and one that's commonly used in Europe is called the Pro-PKD score. And this is just an example of what it looks like. So there's various components, including your gender. Obviously, can't control that, but things like early onset hypertension, and then what kind of genetic mutation you have. And just, I'm going to throw this out there, but just within the last 2 weeks, actually, was an entirely new model that was published in the medical literature. It is, I think, still being understood and digested by nephrologists, and probably needs to undergo some important validation. But this was derived from more than 15 years of data from these 2 landmark NIH-funded studies. And I want to highlight that these were studies funded by the federal government, and which looked at the progression of disease and tested novel treatments. And now the data is still being used years after they're finished to identify a new risk score, which it's hopeful will be more complete or comprehensive than these other ones. So, as Chris mentioned, one advantage of or one approach to studying new treatments for PKD is to repurpose existing drugs, and that's actually what happened essentially with tolvaptan. So again, by repurposing, what I mean is that these are drugs that are already approved by the FDA for conditions other than ADPKD, but they act on biologic pathways that are known or believed to regulate cyst development and cyst expansion in PKD. And at the risk of entirely intimidating people, including scientists, This is a figure from a recent review of the intracellular pathways. And again, just the take home here is that there's many complicated pathways and many individual intracellular molecules involved in these pathways. But what's written in the black font there are known drugs, either drugs in development or drugs already on the market that can affect these. And so this has led to again, approach where you can take these drugs, say, oh, well, I know that this exists, I know this drug affects this pathway, and let's test it. The other advantage is, again, we don't have to go back to phase 1 because we know, at least in healthy people, that these drugs are safe and tolerable. And we have a good sense of what the side effects are already from their use, and sometimes in millions of patients over many years. And that can allow us to develop a clinical trial specifically in PKD, thinking about who might be at the lowest risk of those side effects. So this is in no way intended to be comprehensive, but instead to highlight 4 classes or 4 specific types of medications that are already on the market, already being used, but are being or have been or will be repurposed for testing in ADPKD. Perhaps many of you have already heard about metformin. It's been in the news a lot for all kinds of things, including to slow aging. It's already used for type 2 diabetes. In mice that have genetically induced ADPKD, it leads to a number of benefits, including lower blood pressure, smaller cyst expansion, and better kidney function. We were part of a phase 2 clinical trial that we completed a number of years ago with federal funding. So this is a drug that's generic, so the drug companies are not going to be paying for the studies. And we've showed that it was safe and tolerable over a 2-year period in people with ADPKD, often with advanced disease. And there's now a phase 3 trial that's underway internationally, unfortunately not yet in the US, but that is hopefully going to give us some very important information. SGLT2 inhibitors, which I'll talk about in a little bit, these are drugs that are already approved for a number of conditions, including other forms of chronic kidney disease. In mice, in some studies in mice, it's shown to reduce cyst enlargement. And I have the great pleasure of, with a collaborator, of leading the first in PKD study, in phase 2 trial that's going to be ending in next March. And there are larger phase 3 trials. I'll show you that in a couple slides. I think most people have probably heard at this point about the GLP-1 receptor antagonists like Ozempic. Probably you've heard mostly that they suppress appetite, but they were first developed to lower blood sugar in diabetics and then found to have all kinds of other benefits, including in kidney disease in diabetics. And there's some early studies going on there. And then we'll talk about bempedoic acid, which is actually a lipid-lowering drug, which we're about to start testing as well. So let me move on to talk a little bit more about the SGLT2 inhibitors, and these include drugs like Farxiga and Jardiance that are often advertised on television. And they block the reabsorption of sugar, glucose, and sodium, salt, in the tubules of the kidney. And through a large variety of processes that are kind of still being worked out, they have a number of beneficial effects on the kidneys, including reducing scarring or fibrosis, lowering blood pressure, and decreasing specifically the pressure inside the filters of our kidney, which over time leads to less damage over a period of years. So again, among patients who have chronic kidney disease but don't have ADPKD from other causes, they have it, it lowers the risk of kidney failure and progression of their kidney disease, and also lowers cardiovascular disease. And this is one example of a study done in over 4,000 people who had chronic kidney disease, not from PKD. And the placebo group had a 14.5% likelihood of developing kidney failure or kidney disease progression or cardiovascular death or events. And that was in contrast to about 9%, so about a little over 5 and change percent lower if they got the active drug. And there were multiple other benefits as well. And so this drug, this was Farxiga, is now approved for chronic kidney disease. But they're not approved for use in ADPKD. In fact, patients with ADPKD were excluded from these landmark trials involving 7,000, 8,000, 9,000 patients. And the current treatment guidelines in PKD actually argue or advise against their use for the purpose of slowing progression. But we're starting to get more data about some of their effects in PKD. This data is still very premature and incomplete. An example is one study that looked at the large Veterans Administration database in people who had ADPKD and also happened to have diabetes, and therefore were given these drugs for diabetes. And what it found was that the rate of decline of kidney function, again, that's shown on the y-axis, which was that function was declining as expected in the 9 months before they started the drug, but after it seemed to stabilize. Again, it didn't make their kidney function better, but that's not what we would necessarily expect. But it does seem to slow or even possibly prevent the decline of function. This is not a randomized trial, so these are just some people who got the drug and some people didn't. And so there are potential sources of bias and other explanations. In addition, there is a small crossover study which looked at people already on tolvaptan, and it randomly assigned them to Farxiga or not, and then switched their order of their drugs. And you could see this is, again, the estimated GFR or kidney function over time seems to get better in that dark line, which is the active drug, versus the dashed line, which is placebo. The benefits seem to appear after about 3 months, and they only studied it for another 3 months. So all of this is at least provides a sense of a signal that these drugs may be impactful in people with PKD, but we still need the results of longer term clinical trials, which is what we're going to be getting. So as I mentioned, I'm involved in this trial. I'm one of the co-leaders of the study, and we are studying 50 people using Jardans or empagliflozin and looking at safety and tolerability, but also kidney function and kidney volumes. And you can see that there are a number of larger studies involving now 400 plus patients. Those are being conducted in Europe, and they have not started enrolling yet, but they'll be studying a larger number of patients to really look at whether they can slow progression. So we're hopeful, at least as of March, plus a couple months to clean the data and analyze it, that we'll have the first data in the US in PKD patients, whether this commonly used drug can at least be tolerable and safe, and a sense of whether there's a suggestion of it being efficacious. So I want to move on to another repurposed drug, which in this case is bempedoic acid. And this is a drug that is already approved for people who have high cholesterol in patients who can't take the more common cholesterol-lowering drug like statins, so things like Lipitor and Zocor, or they're taking statins, but their cholesterol is still not high enough. So why are we interested in it? And why is anyone interested in PKD? Well, it acts on an enzyme in the cells lining our kidneys that is important in the regulation of polycystic kidney disease and in the growth and expansion of cysts. One advantage of this drug is that it's only activated in the liver or the kidney, well, those being the organs, of course, and where cysts are most prominent in polycystic kidney disease. And in mice with this disease, with PKD, it seems to slow the expansion of cysts and reduce scarring and fibrosis. And I've just given a quick picture. These are mouse kidneys. And all of these mice have polycystic kidney disease. Some of them got the control or placebo, and some of them got the drug that's BA. And I think you can see in the bottom two pictures on the left is the mice that got no treatment, and they have enlarged kidneys, and all that clear space in their kidneys are cysts. And to the right of that immediately is the mice who got bempedoic acid, and at 120 days, their kidneys are much smaller and have much fewer cysts. And the same thing was seen in mice that were also given tolvaptan, so it seems to potentially be additive to tolvaptan. So we are in the process of studying this with generous support from the Department of Defense. This is a 4-year study. Multiple centers are involved, and it is placebo-controlled and double-blinded. And now that we know what that means from Chris's education of us, and we're going to be looking at, as I said, there's 3 clinical sites in the United States. We're going to be looking at bempedoic acid, 1 pill a day for 2 years, comparing it to placebo. Neither we nor our participants will know if they're getting active or placebo. We're looking at people who have these characteristics, so they have to be at high risk for progression based on some of those methods that I described earlier. Unique, I think, among this study is that people can be using tolvaptan or not. So that's an opportunity to participate if someone is already on tolvaptan. And they have to have a kidney function or glomerular filtration rate of at least 35. And partly that's because this drug has mainly been studied in people who have GFR of over 30. And this is just a quick figure to show sort of the flow of the study. Though there is initially safety follow-up visits at 2 and 6 weeks, but most of these visits are at 3 months, and quite a few of them can be done remotely with blood work at, say, Quest or LabCorp, and then talking to people on the phone to make sure that they're tolerating the medicine well. So we're trying to reduce the burden on participants, because we know they have busy lives just like everyone else, that they won't have to come to our university research center so often. So that study is just about to start enrolling patients within about 1 to 2 months. Again, we're extremely grateful to the Department of Defense for their research support. The other medication that I wanted to highlight was the GLP-1 receptor antagonists. And again, these are, as you know, medicines that can induce weight loss, and in fact are shown to be highly beneficial in people with diabetic kidney disease, in whom they lower the risk of kidney failure and CKD progression. And that's shown in this figure here, where the orange group line are people who got Ozempic, the black line is placebo, and the y-axis is the progression of kidney disease to kidney failure or just decline of kidney function. And so at every time point, it's less if you got Ozempic. And this drug is now approved for polycystic disease. And my colleague at the University of Colorado, Kristen Novak, is in the process of starting or beginning to enroll in a clinical trial using the medication tirzepatide, which is known as Zepbound. So in the few minutes I have, I'm going to just talk about some other drugs that are not repurposing, but these are drugs newly designed and developed to treat PKD. You may have heard, because this was in the sort of public news a lot, this new drug from a company called Regulus, which is farabursen. I'm sure it's going to have a more catchy brand name once it gets FDA approved, or if it gets FDA approved, I should say. It's an oligonucleotide, not a protein. What that means is it's a small molecule made of the building blocks of DNA, and it's administered by injection. It's not available as a pill. And it's designed to increase the production of the healthy or normal form of the protein polycystin. Polycystin is the protein that is mutated and abnormal in people with polycystic kidney disease. So there was an early phase study which showed that it is effective at increasing the amount of functioning polycystin in the urine, again, suggesting that more is being made in the kidneys, and that it may slow the growth of kidney enlargement compared to placebo. It was an early study, small study, so now they're moving on to a larger clinical trial. Regulus is actually bought out by Novartis, and Novartis which is a massive multinational company with a lot of resources. So we're very excited. They're going to be studying this drug initially for 12 months on kidney volume, and then total of 2 years on kidney function. And this is just a cartoon sort of showing conceptually what would happen with this drug. And another drug that is still has a series of letters and numbers as its name. It's being made by Vertex, called VX407. This is targets the misfolding of the protein polycystin, as I mentioned. Polycystin, this is a drug that is just starting to enroll as a phase 2 trial, again, placebo controlled. And so this is one of the first drugs, or maybe the first drug for PKD to go to human studies, that is really targeting a specific type of mutation. So there are some mutations in which no polycystin is made at all, and there are other mutations in which the polycystin is made, but it's altered and doesn't form the right three-dimensional structure and get to the right place in our cells of our kidneys. And that's the kind of mutation that this drug specifically is targeted to help. And so they're looking at patients who are 18 to 65 and have a known mutation, what are called non-protein truncating mutations. Again, some proteins made, it's just not folded the right way. And you can participate in this trial if you otherwise meet criteria, are not sure of your mutation, and the study will actually pay for full genotyping as part of the research study. Lastly, I want to talk about a class of drugs that are targeting this protein called IGF-1. IGF-1 is short for insulin-like growth factor 1. And this protein is made all over our body, so it has lots of effects in multiple organs. But in our kidneys, it regulates cell proliferation and metabolism. Particularly, it tends to speed up cell proliferation, the replication of cells in polycystic kidneys, and make them grow faster. So the biology is complicated. IGF-1 circulates in the blood, but it doesn't circulate just freely. It's bound to these things called binding proteins, and I'll show a picture of it here to help kind of elucidate it. So the binding proteins are shown in the green, and if they're bound to the binding protein, then IGF-1 cannot get to the surface of the cell in our kidney, and it can't induce its effect on prolonging or promoting cyst growth. Now, there is another enzyme in red here called papalysin or papa-A, which actually breaks up this binding protein. So when it does that, the IGF-1 is now free from its binding protein. It's able to get to its cell and exert its adverse effects in polycystic kidney disease. Papalysin is markedly increased in its concentration in PKD, and it seems to be a key mediator of cyst progression in this disease. So what happens when you block papalysin? Well, one of the things that happens in mice is that if you have a PKD mutation that is shown on the very right kidney there, if you block or knock out the gene for papalysin, kidneys look pretty much the same type as that non-PKD kidney on the far left. And if you block it with a medication as opposed to genetics, you also get smaller kidneys as shown in the rightmost kidney compared to the PKD kidney without the drug in the middle. So there are several companies. I've highlighted only two. I apologize if I left some out, but there are several companies that are now have developed novel medicines to block papalysin, and they're going to be tested very soon in phase 1b or phase 2 trials. Some of these trials are actually in the process of launching already as we speak. So I will stop there, and I will now turn it over to Ms. Tasha Vrasper. Thank you.
Hello, everyone. Thanks so much. Thank you to the doctors for giving us all that fantastic information. Again, my name is Tasha, and I actually will be celebrating my 1 year of kidney transplant tomorrow. So I have a lifetime experience of PKD with as a patient. So today I'm going to start by sharing a short bit about my involvement with research throughout the years, and then also share more about how you guys can get involved as patients, advocates, caregivers, and everything like that. So why research and what it looks like? So everyone has a different reason for wanting to participate in research. Mine in particular, I have a long family history of PKD. I watched my dad when I was growing up with PKD, my grandma, my aunt, and then I found out I had it. And that kind of really triggered something in me to want to be motivated to try and make a difference somehow in something that I knew I didn't really have a lot of control over in my life. So I started out getting involved with, as many of us had already talked about, clinical trials, which seems to be the most common type of research that people are aware of. I got involved with that in 2008, so when I was a freshman in college. And basically, the process for that, kind of what we've already talked about, is you need to meet the eligibility criteria for the specific studies. And then once you do that, you move on to the consent, saying that you're going to volunteer and do this. The great thing about that is that any study I've participated in, you can revoke your consent at any time if all of a sudden you just change your mind and you don't want to do it. So it is completely a voluntary experience, which is really fantastic. I participated in, like I said, a couple clinical trials with various medications. And so the really great part of that is that you get follow-up throughout the whole study. I participated in a 3-year study, and I really got to just get extra personal care that I wouldn't have otherwise with my general nephrologist. I also gained a lot of different results. And for instance, I got genetic testing for one of my criteria for a study to see if I qualified. And so I just got a lot of different imaging and different testing done that I otherwise wouldn't have done. And probably, I mean, it's expensive otherwise. So I think that's a really positive thing about participating in clinical studies, is that you're constantly being reviewed, constantly being monitored, along with your general nephrologist and everybody else on your care team. So you just get that extra comfort knowing that you have a lot of people behind your corner. I've also participated in some other clinical trials, such as database trials, where gave a blood sample, and this was kind of more familial related. So I was able to participate with my entire family, and we all submitted blood samples. And then it was really cool to see the results because everybody knows PKD is just so different with every individual, even if you're in the same family. And so just being able to see those results, see how things might be different for different family members in the same area. And then finally, I've participated in the questionnaires and surveys as well, such as the ADPKD Registry, which has been really good with just tracking trends in my own health journey, as well as then all of the other participants that are involved in that. And most recently, I know we talked about it a little bit ago, but I did donate my kidneys after I had my surgery as well. And so I feel like that was kind of brought me full circle with the whole clinical perspective because I was able to give back to research, something that I had been growing for 36 years and hopefully being able to make a big significant difference with that. Like I said, the clinical experience, the research experience has been a very positive one for me. Just getting that valuable personal care and knowing that you're making an impact in even a small way. And so now I'm going to provide some additional ways that everybody can get involved. And so we've already kind of talked about a couple of them. So the big thing I want to say is that no matter what your connection is to PKD, whether you're a patient, family member, caregiver, advocate, medical professional, there's always room for any type of connection to get involved with research with PKD. And then regardless of where you're at in your journey with PKD as a patient, if you're newly diagnosed or even post-transplant, there's always different ways that people can participate, not necessarily having to do specific clinical trials. So I have some of those listed below and the links to them. The PKD Foundation has super great sections about research. They can help you find specific clinical trials if you're interested in that to see if you're eligible, information about how to donate your kidneys after they get removed, if that happens for you, more information about the registry, and then, as we mentioned too, the community reaction panel. So that's a great way for any type of person with a connection with PKD to just get involved with providing your perspective as a caregiver or a patient or an advocate to these research companies so they know how to better provide a patient experience and how to better form their studies to fit patient experiences because we're the ones that have it. We're the ones that watch patients go through these things. And so we are the best advocates for how this disease is progressing in real life and in real time. So thank you everybody so much for listening to us today. And I just wanted to say that research is so important. It's how we move forward towards finding a cure for this disease. And any little step forward is any step forward in progress is making a difference. So regardless of what you do, the important thing is that we are on our way to finding a cure. So now I'm going to pass it on to Paul for some finishing remarks. Thank you so much.
All right. So we are now ready for our question and answer. And we've got lots of great questions that have been submitted. We'll go ahead and start with the first question. This question is for you, Chris. This is from Joan, and she says, if I've been post-transplant for 15 years, if I submit my data to the registry, is that still helpful?
Yeah, absolutely. And I think this is something Tasha hit on, like really any stage that you are in your PKD journey, that data that you're sharing with the registry is super helpful. So you can think about, there are a number of people here interested in the clinical trials associated with transplant and the reduction of immune suppression drugs, but also I think there's a lot of interest, since Tasha mentioned nephrectomies, like following patients long-term, whether or not a patient had a nephrectomy, single nephrectomy, double nephrectomy, no nephrectomy, how that nephrectomy was performed. All those things are data that we can use to determine like what is best practice in the future. So please share your data. It's all very useful.
Perfect. This is a question. Thank you, Chris. This next question is, I think, combined question for Stephen and for Tasha here. if somebody wants to find a clinical trial that fits them, that's right for them, and they have specific criteria, what's the best, how should they approach that? For example, there's one person that says here, well, she wants to participate in a clinical trial where prednisone and rejection medications are not needed for transplant. For example, that's just one example. There's a number of similar kinds of questions. What would you recommend that they do to start figuring out what's available and if it's a good fit? And maybe I'll start with you, Dr. Seliger, and then we can move over to you, Tasha.
Sure. Yeah. No, thanks. That's a great question. So yes, I think the PKD Foundation is absolutely, and there's a list of active studies and protocols and clinical trials, and they also have information on location of some of those studies. And so that would be, I think, a great place to start because those are trials that would be certainly specific to PKD. So there is a website maintained by the federal government called clinicaltrials.gov. And I don't think I included a link into mine, but my talk, I'm sorry. But so you can, and this is a website designed both for scientists and physicians and also for patients. So there's sort of a patient view and a researcher view of it. And you can type in, it's got a fairly sophisticated search engine. You can also limit it again to location, state, country, et cetera. And it'll tell you also whether this trial is yet, maybe it's designed but not yet enrolling, whether it's currently enrolling or whether it's completed. Yeah, thank you. Thanks, Nicole. So that's another great a website to look at and with a very robust search feature.
And that website tells you where to go if you're interested, who to contact too, correct?
Absolutely. Okay. So it has the phone number, email of the investigators, and if it's a multi-site style trial or study, it'll list each site. You can find the one closest to you and then contact those people. And actually, that's how we've gotten contact for almost every trial we've done. We've had people reach out to us through that mechanism.
Anything else you'd like to add, Tasha?
Yeah, just another great way to do that is getting your care team involved. I did that with my nephrologist. I told him, hey, I'm interested in research. If you know anything coming up, please keep me in mind. And so he was able to work with our local research facility here and kind of get my name on the list and so to pass that down to me whenever something came along that I might qualify for as well.
Okay, great. Thanks, Tasha. This next question is just generally about, I think this is a good question because it's it may be sort of a myth or a misconception or a fear about clinical trials. I think I'll start with you, Dr. Seliger. Kaylin asks, if I were to take medication, does that eliminate me from being able to participate in a clinical trial in the future?
Yeah, so I think, you mean, if I understand the question correctly.
Yes, if they're for example, if someone's taking tolvaptan now,
right.
Would they be able to participate in a trial in the future? And I think that goes to the inclusion-exclusion criteria and that kind of stuff.
Yes, absolutely. I think so. So each trial will have its own set of eligibility criteria, including other medicines that one may or may not be taking. So I mentioned our upcoming study of bempedoic acid. So we're opening it to people who are using tolvaptan or people who are not. We're going to try to make sure we have equal numbers of both so we can see. Often with the new experimental medications, the sort of last three that I highlighted, those are almost always not going to be tested in people who are already on tolvaptan. But if someone's been on it and then came off it for whatever reason, they almost always will still be eligible as long as they've been off it for a period of a month.
Period of time.
Yeah.
Yep. I was recently having this conversation with an industry partner, and they were saying, well, it's 6 months. You have to be offered for 6 months. So that may be one of the. All right. Thank you. Thank you, Dr. Seliger. So this is a question, I think first, the next question is first for you, Chris, and then I think a follow-up for you, Tasha. This is a question about donating polycystic livers and polycystic kidneys. The first question is, does the foundation take polycystic kidneys and polycystic livers or donations, biological donations? And that question is for you, Chris. And then maybe Tasha, you can talk a little bit about how you donated yours. So first to you, Chris.
Yeah, so the foundation actually does not have a process for intaking livers at the moment. So I would encourage you to maybe reach out again to your particular clinic and see if they have their own biobank. And so, there still are opportunities for research locally without going through the foundation's current pipeline for donating tissues. But as Dr. Seliger mentioned, he's part of what's called the Research Consortium, which is where the donated kidneys, like the ones that Tasha donated, end up going to. So those are not housed at the foundation, but it goes to kind of the biobank that's housed at University of Kansas.
Okay. And how did you go about donating your kidneys, Tasha?
Yeah, so there is a link on the PKD Foundation website that says, here, fill out this form if you're interested in donating your kidneys. So they do need advance notice. I believe it was like 2 to 3 weeks advance of the surgery. There's 2 forms I filled out. I had to fill out information about my diagnosis, family history, that kind of thing, and then a consent form. And then I had to give those to my transplant center. And then they worked, and I also emailed them to the representative of the foundation, and they worked together to coordinate that because they had to make sure they were in a specific condition and everything when they were moved and packaged properly and all of that. And then, then they did go to Kansas, and the representative was really great. I was able to find out, that they received them and all that. So it was a really nice process.
Great. Thank you. This is an interesting question, and I've never sort of heard this question before. And this is for you, Dr. Seliger. If someone has an adverse side effect early on after starting a clinical trial, do they still need to fully complete the trial?
No,
I think that may be a fear that, yeah.
Excellent question. So, of course, there is always the potential for side effects. We go through that very carefully in the informed consent process. And sometimes for new drugs, the full potential for side effects has not yet been fully described or understood. But there is a general principle called the intention to treat principle in clinical trials, which is if someone has been started in a trial and randomly assigned either to the active drug or placebo, that they continue, even if they stop taking the study medicine, they continue to stay in the trial. And then they have their whatever measurements are being done on their kidney function, their kidney volume, they continue to participate throughout that. Of course, as Tasha pointed out, very importantly, it's always voluntary and anyone can always revoke their consent at any time without any penalty. That's very key. But the person would otherwise continue to participate. And in some cases, people have reactions, and it turns out that they actually were on placebo and the reaction was just a chance thing to happen.
Oh, interesting. Very interesting. All right. Thank you. So this is a broader question and for anybody, for all of you. But Hannah, who's a clinical research coordinator, is just asking about the best ways to reach out more potential patients if you're doing a clinical trial at a center. What are some of the ways that you have seen or have worked with doing outreach to find patients who might be a good fit for your trial? And I guess we can start with you, Dr. Seliger.
Sure. Yeah, I mean, we've used a whole number. So we've used a number of mechanisms. We do at our center, because we've been involved in human subjects research for 15 years. We do have a large registry of people who have already, been to our center for research before and now, are interested in being contacted. We also, because we're in Maryland, as a dense population area, we go and give information to other providers throughout the sort of mid-Atlantic state region so that they can provide that information to their patients. And then we also, we use, of course, the PKD Foundation to help publicize the opportunities for studying for our research studies. And then, as I mentioned before, clinicaltrials.gov. There's many purposes of that, and also for the research community to make sure that the researchers are being entirely transparent with their research. But it does also have the added benefit of allowing patients to find a study that they might be eligible for.
Great. So Chris, Tasha?
Yeah, just to add on, this is really one of the main focus, uses of the registry is to identify people, particularly match people who are interested in learning about clinical trials to clinical trials that they qualify for that are in their geographic region. So if in the case of this particular person asking the question, I would encourage that person to reach out to the foundation, and we can work on ways to use the registry to help recruit for that trial at that particular site.
Great. Any other thoughts, Tasha?
Yeah, I think that just kind of getting community involvement is really important as well. Like we participated in our first walk this year and we had a research booth there and just kind of getting it in front of the patients that are there. We have community meetings that we do as well where we try and do presentations and things like that. So yeah, just kind of really trying to get it in front of people. I think in person as well has been successful.
Thank you. Thank you. Next question is from Kaylin. Let me just ask this question. You may or may not be able to answer it. She asked about a study called the Freedom Study, which was a kidney recipient received stem cells from a donor with little or no transplant medication. Are you familiar with that trial? Is it still going?
Yeah, I'm sorry, I'm not familiar with the trial,
Okay.
and my research has really been in general nephrology and PKD. There's a lot of research going on in transplant of novel immunosuppressive therapies,
Got it.
including stem cells. But sorry, I can't get
Yeah.
more.
Yeah, so the
Maybe.
foundation actually promoted that trial because the news was just a couple of weeks ago that it came out.
Oh,
I can, since we have the information for the person that asked the question, I can send the link to the trials, but it is listed under clinicaltrials.gov. So you could probably Google search it, but I also will make sure to respond back to that.
yep. So we'll have that. Yeah. So thank you. Thank you, Chris. So this is a question, Chris or Dr. Seliger. How is TKV measured, and what is the Mayo imaging classification system? So maybe I'll turn this over to Dr. Seliger
Sure.
for this.
Yeah. So TKV is measured with abdominal imaging. The most common and method of abdominal imaging that is used for this is MRI. It doesn't involve any IV contrast, so it's just an abdominal MRI scan. But it can also be quantified using CAT scan, and there are some ways to also do it with ultrasound, although MRI certainly provides the most accurate and precise measures. It's just exactly what it sounds like, which is the total volume of each kidney summed together. I think what makes it maybe a little bit harder to use clinically is that if you go to a regular chain radiologist and you have your MRI performed, it actually takes a lot of added time to do those measurements of about 20 to 30 minutes. And that's usually time that your insurance is not going to pay the radiologist for.
Got it.
So yeah, there are various ways now we're trying to use artificial intelligence to make it much quicker. And again, there's almost every research study will perform it as part in a standardized fashion as part of a research protocol. So.
Got it.
If I can just add, so the RRC just incorporated a new imaging core. It's out of the Rogosin Institute, and the radiologist there has a platform for anybody to go and upload their imaging data and get a TKV value if you're interested. So that website is traceorg.com, TRACEORG.com. So a resource that may be useful.
Great. We've got time for two more questions here. The next question I'm going to turn over to you, Chris, because it's specifically about the registry. And this person is concerned about the overall time commitment and was wondering, what is the time commitment if you submit a profile to the registry, and the amount of time and effort that is involved in submitting their profile and then updating it. If you could talk a little bit about that.
Yeah, I think it only takes a matter of minutes to actually sign up for the registry, go through the consent forms and participate. And then beyond that, the amount of time is really dependent on the actual participant. I would say one of the most valuable things you could do is link your electronic health records, and then those will be automatically updated and kept up to date. You don't have to do anything, but all that data is available for research. It also allows you to be alerted to clinical trials that you might qualify for. And there's really no effort on the part of the participant. If you want, you can use it to actually function as almost like a journal, logging your own personal journey, like pain scores and impact on quality of life. Those kinds of things can be like self-reported, so that when you go to your physician, that you can show kind of longitudinal data that you've recorded over time. And that's something that, if you want to use the platform for that purpose, it takes more time, but I think it also has a lot of value for people. So it's the spectrum. You can put in as much effort as you want, but really just linking your electronic health record, stepping away is really, really a valuable source of information for the registry.
Can I add something?
Yeah, please. Yes, Tasha, I was going to say that. Yes,
Yeah. I
personal
just wanted to
experience.
say too, the PKD Foundation does a really great job of sending out reminder emails. And so a lot of times I just totally forget about it. And then I'll get an email like, oh, hey, take this survey to update your pain score or update your personal history or anything like that. And so it's not like it times out or expires or anything. You just get notifications like, hey, if you have time, update this. So it's really nice and convenient and a reminder, because otherwise, I mean, we don't need to worry about it.
Got it. Yep. Thank you. So this is going to be our last question, and I think this is probably one that is on a lot of folks' minds, and I think it comes with a little bit of frustration, but it also mean you also have to understand the process that it takes to get to a new treatment. So the question is, why is it taking so long to find a cure for PKD? And I think it's a complicated answer, but I thought I would pose that as a final question for any of our panelists here to answer and pitch in on. So, well, we can
I would
start with you,
say,
Tris, if you'd like.
as Steve mentioned, I think I take a less pessimistic look at this and super optimistic. I look at the new therapies that are coming along in the clinical trials that are just starting to enroll here in later stages. And some of these do have the potential to be curative. I know Steve didn't have an opportunity to really go into the detail of it, but some of these are therapeutics that get at the real heart of what causes cysts to form. And there's data out there that shows that cyst development is reversible. So not only can we potentially stop cysts from forming, but in these preclinical models, we're reversing back to more normal kidney phenotype. And I think that's super exciting. And so I understand people who, I can't 100% relate to people who have, lived with this disease, like maybe Tasha has, but I can understand that being frustrating that we only have tolvaptan to date, but it's really a new time in terms of research. And I would encourage any person who's frustrated, reach out to the foundation so we can try to explain some of what's new and exciting, and encourage those people to become involved. I think the opportunity to be involved in a potential cure for this disease is like through participating in a clinical trial is something that is a really great opportunity, and something that people can be proud of being part of.
Fantastic. Dr. Seliger or Tasha, anything else you'd like to say about that topic?
Yeah, I guess I would share Chris's enthusiasm for where the science is going. And, it's in my professional career, I can remember when the gene for PKD was first actually described. And I'm not quite that old. So to go from there to now, where we have drugs that are targeting that whole genetic mechanism, even in the time frame that we've seen, that actually represents, to my mind, some really exciting advances over what is actually a relatively rapid period.
All right. Well, fantastic. Well, thank you all, Chris and Dr. Seliger and Tasha, for your wonderful presentations and answering so many great questions. Thank you all for submitting so many great questions. We're honored to have had this opportunity to learn from all of you on this important topic in PKD. With that, this concludes today's program. Thank you all.